Hi everyone. We reviewed Chapter 6, Transcriptome and Proteome Analysis, but we did not finish the last part of protein interaction assay. There are several different ways of identifying protein-protein interaction. We reviewed the proteome analysis to review the expression profile of the proteins at certain occasion. But even if those proteins are expressed, actually mostly the proteins function as a whole protein complex and physical interaction with associated proteins. The protein-protein interaction provide information how the protein functions in the cell. We first reviewed the identifying pairs of interacting proteins, and we focused on review yeast-2 hybrid system. Identifying components of multiple protein complex, there's affinity chromatography and co-immunoprecipitation. I put up the video tutorials of yeast-2 hybrid system and affinity chromatography and co-immunoprecipitation on Moodle, so make sure you review those. The major limitation of yeast-2 hybrid system is that only if those two proteins have direct physical interaction, you can determine those two binding partners. For example, if there are multiple proteins associated as a complex, and if you are looking for the interaction of the one red and the orange one, which does not have direct physical interaction but have an interaction with a large protein in the middle so that it forms the entire complex, you can't really determine those two proteins function in the same complex by yeast-2 hybrid system. Some of the peptide does not interact with all members of the complex, so this is the major issue with the 2 hybrid system in determining the protein-protein interaction. So then what other method can be utilized to determine the entire protein complex, including those proteins does not have direct physical interaction? One is affinity chromatography, which is used for purifying the multiple proteins. As long as you can extract the protein without disrupting the entire protein complex, if you use one as a bait that has an interaction with one of those proteins, then you can pull out the entire protein complex using affinity chromatography. You may had a chance to learn about affinity chromatography in chemistry, basically separating some components from the mixed population by using two molecules that have strong affinity to each other. For example, in this case, let's use some of the antibody binding to one of those proteins, making a complex. You fill the column with antibody, and you're going to run the entire cell extract. If there's any protein has a strong binding affinity to the antibodies, basically the protein that has a specific antibody raised, then the protein will be binding to the antibody in the column. So that first step is eliminating those proteins that are not associating with the antibody. Then you're going to wash the column several times so that all the unnecessary debris are all washed out, including other non-specific proteins. And you are going to change the condition, mostly the condition to maintain the binding of the antibody and the protein are based on the low salt condition. When you increase the salt, the high salt will interfere with the protein antibody interaction so that the protein is now detached from the antibody, released from the column, and drop into the collecting tube. So if you collect those complex protein and analyze what kind of proteins are isolated by the affinity chromatography, then you will see the whole collection of different proteins have a binding complex together. Some proteins have a strong binding affinity to specific divalent cation. This is an example with a protein called calmodulin. Calmodulin has a strong binding affinity to calcium because calmodulin is functioning through the calcium activation. So if you fill up the calcium in the column and run the protein extract through the column, then only if the protein interact with the calcium will get cut. The rest of them will be eluted out. And if you decrease the calcium condition, that used to bind inside the column will now be released and go down to the collection tube. If there's any protein that has a physical interaction with the calmodulin protein, those will be also eluded in here. So by using this column, you can detect the other proteins that has a direct physical interaction with calmodulin protein. So this is one of the good example of identifying the actual protein function in the cell. Affinity chromatography but still have a lot of limitation because sometimes as while you are doing the protein extraction, some protein may be detached from the whole complex. So even if it does not have a whole capacity of identifying the entire protein complex, but still this affinity chromatography has a lot better approach in identifying multiple proteins binding a single complex. So then, why do we study the protein-protein interaction? The ultimate goal of doing a protein-protein interaction study and protein profiling is to determine how the proteins are associated and how they actually govern function of the cells. So they want to know the functional interactions between different types of protein complexes. For example, you have a respiratory molecules that digesting glucose and generate energies. During the glycolysis, there are multiple proteins function in the pathway, and once it goes through electron transport system, there are additional proteins function for this process. So even if one governs one biochemical pathway, the other one governs the other pathway, but ultimately these protein pathways are associated together. By studying those protein-protein interaction study, you can develop a flow of how the protein complexes have an associated proteins. Or sometimes there are regulatory modules that interact with the multiple pathways to regulate those pathways function. So you can see what kind of protein complexes are the regulatory molecules, what kind of proteins are actually the functional molecules, and how they are networking together. That's basically what the scientists try to do by protein network assay. So the protein interaction map display the entire protein-protein interaction. So we call those entire map as an interactome. This is one of the earliest version of protein interactome network developed by the Saccharomyces cerevisiae proteins that have identified and their protein interaction was discovered. As a simple example, let's say you see there's a one core and multiple proteins have a connection together. These are the entire protein complex function and regulated by the center molecule. And those different color indicates different enzymes or proteins that have different functions. And if you look at those different modules that having a small physical interactions, those are having a more complex network with other protein molecules, and eventually those are all connected as a whole interactome. This is more detailed protein interactome assay, and each circle indicates different numbers of proteins that are associated together. So one interactome has 183 proteins interact together as a whole complex or individual molecules, and some of them has a much less proteins associated together for a simple function. As you can see, those oval shape is the protein complex, so which is formed as a multiple proteins. And among those entire protein complex, you can determine what complexes governs different cellular function. For example, the red color protein complex governs the cell cycle related function. Dark green governs the function related to signaling network. Blue protein complexes govern the transcriptional regulation and DNA maintenance. Pink proteins govern transport. Orange color governs DNA metabolism, etc. So if you look at the complete interactome network, you will see some of the distinctive features in the protein network. Some are having a very small complex gathered together, and some others have more association related protein interaction together. And those gathered spots that have multiple proteins interact together we call hub. Especially the hubs are in two different kinds, depends on how they are associated with other protein molecules. For example, the party hubs are those networking within the individual biological processes and does not contribute to the overall organization of the proteome. For example, respiratory protein complexes is party hub that governs individual biological process. Fermentation protein complex is individual party hub governing individual biological process. If there is any protein complex governs the biological processes of fermentation, respiration, and other glucose metabolic pathways that we call as date hub. Date hub is the key player that provides the organization of the entire proteome by linking individual biological processes. Shown here provides some examples of what will happen if we remove the party hubs. So I mentioned that the party hubs are individual biological processes governing different cellular functions. So remember, the original protein complex network has called the protein complex. The original picture of the complete network looks like this, have connections, and there are individual area where you can see those each biological processes are. So if you remove the party hub, you will see those individual hubs are gone. And if you remove the date hub, which is the linking of individual biological processes, then you will see those individual functional hubs are still remains, but the connection of each biological functions are eliminated, so they are kind of scattered as an individual processes. So we have reviewed genome, transcriptome, and proteome analysis through the genome sequencing project and annotation, and several methods to study transcriptomes, and several approaches to study proteome. So using those knowledge from genome, transcriptome, and proteome analysis, we can better understand the entire gene expression and their function in governing cellular processes. So those information provide good insight in understanding biochemistry of the cell and how the entire cell system function in terms of what kind of proteins are involved, how they are regulated, or how they are associated to govern the entire metabolic pathway of the cell. Beyond the proteome analysis is to understand the entire regulatory mechanisms by the environmental change and the environmental signals come to the cells. These are some of the signaling cascade you have previously understood. The protein regulations are sometimes governed by the receptor ligand association, and those protein complexes actually bind with the DNA and interact the gene transcription. And sometimes those protein complexes interact with the protein translational machinery so that the protein synthesis are regulated. This is one of the example of the glucose sensing network and receptor detecting the glucose, and it actually transferred the signal to multiple proteins, including the cell cycle regulating proteins. HDA1 and SWE1 complex is involved in histone modification, and HXK2 is the protein actually express the hexose transporter and hexose metabolic proteins, and MYC1, MYC2 is another regulatory molecules that actually bind and regulate the entire protein complex. What I can tell you here is that there are multiple regulatory proteins affect on the same proteins by different signals. So if there's one signal comes, then this will activate those protein molecules to rearrange the genome structure to activate some of the functional molecule. And in response to the other signal, different protein is activated and regulate similar proteins. and so does the another protein that responding to different signals can actually activate and regulate those entire protein complex. So the key take-home message here is that when the protein regulation occur, there is not a single pathway either activate or inactivate the entire protein complex, but there are multiple, sometimes redundant, cross-talking between those regulatory proteins depends on those environmental changes. Even if they are responding to same glucose concentration in the environment, there are at least 3 or 4 different pathways responding the glucose signal simultaneously. And if one failed to deliver the signal, then the other pathway can overcome and then deliver the signal so they turn on the same sets of the genes in response to the glucose sensing. So as a concluding remark of the entire lecture from 1 to lecture 6 is that with the entire genome, transcriptome, and proteome analysis, eventually we will study metabolome, which is the mechanisms by which those protein expressed from the major genome governs the cellular metabolic pathways. All the informations are stored in genome as a blueprint, and then those blueprints are expressed differentially depends on the environmental condition and developmental status and response to different signals. And understanding those metabolome is important for understanding human disease because a lot of cases those malfunctioning or out of regulation of those protein processes result in disease development. Understanding those metabolic pathway and their regulatory processes of the disease development can be a fundamental basis of developing drug targeting the specific disease.